This video contains audio content only. There are no visual elements or images accompanying the audio. If you're expecting to see video footage, please note that this is an intentional audio-only presentation. We recommend listening to this content rather than watching the screen. Thank you for your understanding and enjoy the audio. Welcome to an exploration of one of the most captivating narratives in the history of life on Earth, the evolution of mammals. This journey, spanning hundreds of millions of years, is a testament to the power of adaptation, the intricate interplay between genes and environment, and the remarkable diversity of life that has emerged on our planet. In this easy-to-understand explanation, we will unravel the mysteries of mammalian evolution, tracing their lineage from ancient reptilian ancestors to the diverse array of furry, warm-blooded creatures that inhabit our world today. Prepare to delve into the deep past, venturing back to a time when the Earth was dominated by giant ferns and towering lycopods, and the first whispers of mammalian life began to emerge from the shadows of their reptilian predecessors. We'll uncover the secrets of the synapsids, the stem mammals, and trace their rise and fall, setting the stage for the emergence of true mammals. We will explore the pivotal transitions that shaped mammalian form and function, from the evolution of an erect posture and specialized teeth to the remarkable transformation of the jaw and ear. This journey will take us through the Mesozoic era, the age of dinosaurs, where mammals, though small and often overlooked, continued to diversify in the shadow of their reptilian overlords. We'll discover the surprising diversity of Mesozoic mammals, from semi-aquatic creatures with beaver-like tails to gliding mammals that took to the air. We'll then witness the rise of mammals in the Cenozoic era, following the extinction of the dinosaurs, and explore the factors that fueled their remarkable diversification and the emergence of mammalian giants. But the story of mammalian evolution is not just about bones and teeth. We'll delve into the hidden world of genes, exploring how molecular phylogenetics has revolutionized our understanding of mammalian ancestry, revealing the intricate relationships between different groups and the timing of their divergences. We'll also explore the fascinating co-evolutionary tale of mammals and their gut microbes, a symbiotic partnership that has shaped digestion, immunity, and even behavior. The evolution of mammalian reproduction, a story of remarkable innovation, will also take center stage. We'll explore the evolution of the placenta, a defining characteristic of placental mammals, and its profound impact on maternal evolution and reproductive strategies. And we'll delve into the mysteries of the mammalian brain, tracing its remarkable transformation from the olfactory world of early mammals to the complex cognitive abilities of modern humans. Finally, we'll explore the social fabric of mammalian life, examining how cooperation, communication, and group living have shaped their destiny and contributed to their evolutionary success. From the cooperative vigilance of meerkats to the intricate songs of humpback whales, we'll discover the diverse ways in which mammals interact with each other and their environment. By the end of this exploration, you will have a deeper understanding of the remarkable journey of mammals, from their humble beginnings to their current dominance in the world's ecosystems. You'll gain insights into the key evolutionary transitions that shape their form and function, the genetic and molecular underpinnings of their diversity, and the complex interplay between ecology, behavior, and evolution. Prepare to be amazed by the ingenuity of evolution and the incredible story of mammalian life on Earth. Chapter 1. From Synapsid Shadows, the Dawn of Mammals. Our story begins, not with the familiar furry creatures we call mammals, but with their more ancient, reptilian-like ancestors, the synapsids. Picture the Earth during the late Carboniferous period, over 300 million years ago. Giant ferns and towering lycopods dominate the landscape, and among them, a new group of vertebrates is emerging. These are the synapsids, sometimes called stem mammals, or proto-mammals, distinguished by a single opening in their skull behind each eye, the temporal fenestra. This seemingly small feature sets them apart from the sauropsid lineage, the evolutionary path that would eventually lead to reptiles and birds. The Permian period, following the Carboniferous, witnessed the synapsids rise to dominance. They diversified into a remarkable array of forms, filling ecological roles as both top predators and large herbivores. Dimetrodon, with its distinctive sail back used for thermoregulation or display, is perhaps the most iconic example. But the synapsid dynasty included a far greater cast of characters. Imagine Cotylodontus, a massive herbivore reaching lengths of up to 6 meters, lumbering through the Permian forests. Or the agile and fearsome Sphenecodonts, a group of carnivorous synapsids with powerful jaws and sharp teeth, preying on the abundant herbivores of the time. The synapsids' success is reflected in their skull structure. The temporal fenestra, that single opening behind each eye, not only lightened the skull but also provided crucial attachment points for jaw muscles. This allowed for a wider range of jaw movement and a stronger bite, giving them an edge in the cutthroat world of the Permian. However, the synapsids' reign would not last. The Triassic period brought with it a new challenge: the rise of the archosaurs. This group, characterized by a more upright posture and superior locomotion, included the ancestors of crocodiles, pterosaurs, and dinosaurs. The archosaurs' competitive advantages were numerous. Their upright posture allowed for greater stamina and faster movement, while their more efficient respiratory systems provided them with the energy needed for an active lifestyle. The synapsids, faced with this new competition and a series of environmental changes, including volcanic eruptions and fluctuating temperatures, began to decline. Within the synapsids, however, a smaller, less conspicuous group was evolving: the mammalia forms. These creatures, facing the daunting challenge of archosaur dominance, adapted to a nocturnal lifestyle. This shift to the shadows of the night had profound implications for mammalian evolution. One crucial adaptation was the development of a bony secondary palate. This structure, separating the nasal passages from the mouth, allowed mammalia forms to eat and breathe simultaneously, a crucial advantage for sustaining high metabolic rates, possibly hinting at the early stages of endothermy. The nocturnal world also placed a premium on senses other than vision. The sense of smell became increasingly important, driving the evolution of a larger brain and more complex nasal structures. Evidence for this can be found in the shapes of fossilized teeth, indicating changes in diet and feeding habits, and in the development of turbinates, thin bony structures within the nasal cavity that increase the surface area for detecting scents. The nocturnal lifestyle had further implications for the evolution of endothermy and hair. The darkness offered a thermal advantage, as mammalia forms could avoid the scorching heat of the Triassic sun. However, the cooler night temperatures also presented a challenge, driving the need for insulation. Hair or fur, providing this much-needed insulation, became a key mammalian feature, helping to maintain a constant body temperature even in fluctuating environments. The Mesozoic era, often called the Age of Dinosaurs, saw mammals continue to diversify, albeit in the shadow of their reptilian overlords. They explored new ecological avenues, evolving unique adaptations for survival. Recent fossil discoveries, particularly from China, have revealed a surprising diversity of Mesozoic mammals. Castorocauda, a semi-aquatic mammal with a beaver-like tail and webbed feet, thrived in the Jurassic lakes and rivers. Volaticotherium, a gliding mammal with a membrane of skin stretched between its limbs, took to the air, much like modern-day flying squirrels. And Rhipenomammus, a badger-sized predator, even dared to hunt juvenile dinosaurs, providing evidence that mammals were not entirely relegated to the sidelines during the Mesozoic. These fossil discoveries, combined with the insights provided by molecular phylogenetics, have revolutionized our understanding of mammalian evolution, allowing us to trace relationships between different groups and estimate the timing of their divergences. Throughout this evolutionary journey, other defining mammalian characteristics emerged. Mammary glands, enabling mothers to nourish their young with milk, lactation, evolved, although the fossil record offers limited insights into the evolution of these soft tissues. An erect limb posture, placing the limbs beneath the body rather than splayed out to the sides, provided greater agility and efficiency of movement. Fur and hair, evolving alongside endothermy, provided insulation and protection from the elements. Warm-bloodedness, or endothermy, allowed mammals to maintain a constant body temperature, independent of external conditions, enabling them to thrive in a wider range of environments. The story of mammalian evolution is a testament to resilience and adaptability. From the shadows of the Permian and Triassic, they emerged, transformed by the pressures of competition and environmental change. Their nocturnal origins shaped their senses, their physiology, and their very being, setting the stage for their eventual rise to dominance in the Cenozoic era. Chapter 2. Whispers in the Genes. unraveling the secrets of mammalian ancestry. The fossil record provides a tangible narrative of mammalian evolution, a story etched in bone and stone. But the whispers in our genes offer a deeper, more nuanced understanding, revealing the intricate relationships between mammalian groups and their extinct relatives. Molecular phylogenetics, the science of comparing DNA sequences, has revolutionized our understanding of mammalian ancestry, providing a molecular clock to time evolutionary events and illuminate the branches of the mammalian family tree. Imagine the power of comparing a single gene across diverse species. Take, for example, M6p, IGF2R, a gene crucial for growth and development. In marsupials and placental mammals, this gene is imprinted, meaning only one copy, inherited from either the mother or the father, is active. This intricate mechanism of gene regulation plays a crucial role in balancing fetal growth. However, in monotremes, the egg-laying mammals like the platypus and echidna, M6p, IGF2R is not imprinted. This suggests that genomic imprinting, a sophisticated form of gene regulation, evolved after the monotreme lineage diverged from the marsupial and placental mammal lineage. Even more striking, the M6p, IGF2R protein in monotremes doesn't bind to IGF2, an essential growth factor, a feature shared by other mammals. This difference highlights a fundamental shift in the function of this gene during mammalian evolution. Beyond individual genes, whole genome analyses, examining the entire DNA blueprint of an organism, provide a panoramic view of evolutionary history. Comparing genomes across a wide spectrum of mammals, from the platypus to humans, reveals shared genes, unique genetic signatures, and the telltale traces of ancient genomic parasites. The platypus genome, a remarkable mosaic of genetic information, exemplifies this evolutionary tapestry. It contains genes reminiscent of reptiles, involved in egg-laying, alongside genes characteristic of mammals, such as those responsible for milk production, although platypuses notably lack nipples, secreting milk through their skin. This genetic blend reflects the platypus's unique position on the evolutionary tree, representing an early branch of the mammalian lineage that retains features of its reptilian ancestors. The abundance of repetitive elements in the platypus genome, however, poses a formidable challenge for genome assembly, complicating the task of piecing together the complete DNA sequence. The Y chromosome, a key player in male sex determination, also harbors evolutionary secrets. Comparisons between the human and chimpanzee Y chromosomes, surprisingly, reveal significant divergence in structure and gene content, despite the relatively recent split between these two lineages. This unexpected divergence underscores the rapid evolution of the Y chromosome, possibly driven by factors such as sperm competition and the accumulation of male-specific genes. Studying the evolution of soft tissues, such as mammary glands responsible for lactation, presents a unique challenge. These delicate structures rarely fossilize, leaving gaps in the paleontological record. Genetic data, however, can complement these findings, providing clues about the origin and diversification of mammary glands. Similarly, the evolution of fur and hair, crucial for insulation in warm-blooded mammals, can be traced through genetic analyses, revealing the genes responsible for hair development and their evolutionary history. The evolution of the secondary palate, a bony structure separating the nasal passages from the mouth, is another example of how genetic data can enhance our understanding of mammalian ancestry. This structure, allowing mammals to eat and breathe simultaneously, is crucial for sustaining high metabolic rates associated with warm-bloodedness or endothermy. Genetic studies can pinpoint the genes involved in palate development and trace their evolution across different mammalian lineages. Molecular phylogenetics offers a powerful tool for estimating the timing of key evolutionary events. By comparing DNA sequences and calibrating molecular clocks with fossil evidence, researchers can estimate the divergence times between different lineages. This has allowed us to pinpoint the emergence of diurnal mammals, active during the day, a shift from the nocturnal lifestyle of early mammals. The diversification of placental mammals, a group characterized by prolonged internal development of the fetus, has also been timed using molecular data, revealing a burst of diversification following the extinction of the dinosaurs. The study of imprinted genes, like M6p, IGF2R, provides a unique window into the evolution of mammalian reproduction and development. The imprinting status and function of this gene vary across different mammalian groups. In monotremes, its lack of imprinting and different protein functions suggest an ancestral state. In marsupials and placental mammals, the evolution of imprinting reflects adaptations to specific reproductive strategies. Transposons, mobile genetic elements sometimes referred to as jumping genes, have played a significant role in shaping mammalian genomes and driving the evolution of new traits. These genomic parasites, by inserting themselves into different locations in the genome, can alter gene regulation and function. For example, transposons have been implicated in the development of the placenta, a complex organ connecting the developing fetus to the uterine wall, and in the evolution of venom production in the platypus, a unique defensive mechanism in this egg-laying mammal. The ability of transposons to randomly insert themselves throughout the genome has led to their involvement in diverse evolutionary innovations, from the progesterone-responsive uterus essential for pregnancy to venom production. The study of mammalian gut microbiota, the community of bacteria residing in the digestive tract, also benefits from molecular phylogenetics. By analyzing 16 srRNA gene sequences, researchers can identify the different bacterial species present and trace their co-evolution with their mammalian hosts. These studies reveal the influence of diet and host phylogeny on gut microbial diversity and provide insights into the role of the microbiome in mammalian evolution. The use of nuclear and mitochondrial genes has further allowed scientists to infer behavioral changes, such as the emergence of diurnal activity in early mammals, providing a molecular perspective on the evolution of mammalian behavior. The story told by our genes intertwines with the fossil record, providing a richer, more complete narrative of mammalian evolution. The whispers in our DNA reveal the timing of divergences, the evolution of key traits, and the influence of environmental pressures, painting a vivid picture of mammalian ancestry. From the rise of the synapsids to the diversification of modern mammals, the language of genes offers a powerful lens through which to view the grand sweep of evolutionary history. Chapter 3. Sculpting Form: The Rise of the Mammalian Blueprint. Chapter 2 explored the whispers in our genes, tracing the intricate branches of the mammalian family tree. Now, we turn our attention to the symphony of bones and teeth, the tangible expression of that genetic heritage. The evolution of mammalian form is a story of remarkable transformations, a testament to the power of natural selection to sculpt the bodies of animals, adapting them to ever-changing environments. Imagine the earliest synapsids, their limbs splayed out to the sides, much like modern-day lizards. This sprawling posture, while suitable for their environment, limited their agility and speed. The transition to an erect stance, a defining characteristic of mammals, involved a series of skeletal modifications that unfolded over millions of years. The limbs, once positioned at the sides of the body, gradually rotated beneath the body, bringing the plane of action closer to the trunk. This shift, evident in the fossil record of early therapsids, enhanced locomotion by increasing stride length and efficiency. The shoulder and pelvic girdles, the skeletal frameworks supporting the limbs, also underwent significant changes. The shoulder girdle became more streamlined, allowing for greater flexibility and range of motion in the forelimbs. The pelvic girdle, in turn, became more robust, providing a stable base for the hind limbs and facilitating the powerful thrust needed for running and jumping. This transformation from sprawling to erect posture, clearly visible when comparing the skeletal structure of a reptile like Varanus to an early mammal like Morganeucodon, marked a significant leap in mammalian evolution, paving the way for greater agility and speed. Think of the swift movements of a cheetah, a testament to the evolutionary legacy of this skeletal shift. The evolution of the mammalian middle ear is another remarkable chapter in this story. Early synapsids, like their reptilian ancestors, had a jaw joint composed of the articular and quadrate bones. But in mammals, these bones embarked on an extraordinary journey, migrating from the jaw to the middle ear, transforming into the malleus and incus, respectively. This migration is beautifully documented in the fossil record, with transitional forms like Hadrocodium showcasing the reduction of these bones in the jaw and their increasing prominence in the middle ear. Joining the stapes, a bone already present in the middle ear of earlier vertebrates, the malleus and incus formed the three bone structure characteristic of mammals. This intricate arrangement, a marvel of evolutionary engineering, dramatically improved hearing, particularly of high-frequency sounds. This enhanced auditory acuity provided a crucial advantage in the nocturnal world of early mammals, allowing them to detect the rustling of prey or the approach of predators in the darkness. Consider the acute hearing of a bat, navigating and hunting by echolocation, a direct descendant of this evolutionary innovation. Teeth, the hardest parts of the body, offer an enduring record of evolutionary change. Early mammals transitioned from homodont dentition, where all teeth are similar in shape and function, to heterodont dentition, a hallmark of mammalian diversity. This transition, a pivotal moment in mammalian evolution, unlocked a world of dietary possibilities. Incisors, sharp and chisel-like, evolved for nipping and gnawing. Canines, pointed and formidable, became specialized for piercing and tearing. Premolars and molars, with their complex cusps and ridges, developed into powerful grinding tools. This dental diversification allowed mammals to exploit a wider range of food sources, from insects and fruits to leaves and even meat. Consider the rodent's ever-growing incisors, perfectly adapted for gnawing, or the carnivore's powerful canines, designed for tearing flesh. The evolution of herbivorous mammals, like the giant Indricotherium, saw the development of high-crowned molars for grinding tough vegetation. Even within closely related groups, like the various species of Amospermophilus, antelope squirrels, variations in tooth shape and size reflect adaptations to specific diets and habitats. The evolution of chewing, a defining characteristic of mammals, presented a unique challenge: the need to breathe simultaneously. This challenge was met by the development of the secondary palate, a bony structure separating the oral and nasal cavities. This separation, a key innovation in mammalian evolution, allowed for uninterrupted breathing while chewing, crucial for sustaining high metabolic rates. The zygomatic arch, or cheekbone, provided a sturdy anchor for the masseter muscle, a powerful jaw closer essential for the grinding action of chewing. This muscular arrangement, evident in the robust zygomatic arches of many mammals, further enhanced the efficiency of food processing. The evolution of the mammalian nose, often overlooked, is another crucial adaptation. Respiratory turbinates, thin bony structures within the nasal cavity, dramatically increase the surface area for interacting with inhaled and exhaled air. These structures, like miniature air conditioners, play a crucial role in warming and humidifying incoming air, preventing damage to delicate lung tissues. Equally important, they recover heat and water from outgoing air, conserving precious resources and maintaining body temperature. This efficient system of air processing, a hallmark of mammalian physiology, is essential for maintaining a high metabolic rate and supporting an active lifestyle. The ability to place the larynx in an intranarial position, further separating the airway from the food passage, represents another key innovation. This arrangement, controlled by specialized pharyngeal muscles, allows mammals to breathe while eating, preventing choking and maximizing oxygen intake. This adaptation is particularly evident in species like Pachygenellus, an early mammalia form that shows the first signs of a pterygoid hamulus, a bony projection associated with the development of these crucial pharyngeal muscles. The evolution of the mammalian jaw is a story of simplification and specialization. Early synapsids, like their reptilian ancestors, had a jaw joint formed by the articular and quadrate bones. As we've seen, these bones migrated to the middle ear in mammals, leaving the dentary bone to become the sole bone of the lower jaw. This single bone, articulating with the squamosal bone of the skull, formed the new dentary squamosal jaw joint, a more robust and efficient chewing mechanism. This simplification of the jaw, documented in fossils like Morganucodon and Jeremiah, allowed for a more powerful bite and a wider range of jaw movements, crucial for processing diverse food sources. The fossil record reveals a fascinating array of jaw adaptations in different mammalian groups. The powerful jaws of carnivores, like the extinct saber-toothed predators, reflect their specialized diet, while the elongated jaws of some herbivores, like the early horse ancestors, are adapted for cropping vegetation. The evolution of the jaw, a testament to the interplay of form and function, played a pivotal role in the diversification of mammals into various ecological niches. The evolution of mammalian form is a testament to the power of adaptation. Each skeletal and dental modification, each refinement of the senses and respiratory system, played a crucial role in the survival and diversification of mammals. From the sprawling posture of their reptilian ancestors to the erect stance of modern mammals, from the simple teeth of early synapsids to the specialized dentition of today's diverse mammalian groups, the story of mammalian evolution is etched in bone, tooth, and tissue, a symphony of form sculpted by the relentless forces of natural selection. Chapter 4. The Rise of the Furry and Warm-Blooded, a Triumph in the Cenozoic. The Cenozoic era, dawning after the cataclysmic asteroid impact that ended the reign of the dinosaurs 66 million years ago, became the stage for the remarkable diversification of mammals. No longer overshadowed by reptilian giants, mammals exploded into a multitude of forms, seizing the ecological opportunities left vacant. This remarkable rise was fueled by a suite of physiological innovations that allowed mammals to thrive in a world undergoing dramatic transformations, a world of fluctuating temperatures and emerging ecosystems. Central to this mammalian triumph was the evolution of endothermy, the ability to generate and regulate internal body temperature. Unlike their ectothermic reptilian predecessors, whose body temperature fluctuated with the environment, mammals could maintain a constant internal temperature, freeing them from the constraints of external conditions. This newfound independence allowed mammals to remain active in a wider range of environments and across daily temperature fluctuations, opening doors to new niches and lifestyles. Imagine the cool night air of the early Cenozoic, where small, furry mammals could forage and hunt while their cold-blooded competitors were dormant. Fur or hair, a defining characteristic of mammals, played a pivotal role in this endothermic lifestyle. Evolving primarily for insulation, fur provided a crucial barrier against heat loss, allowing mammals to conserve precious energy and maintain a stable body temperature even in colder climates. This insulation was particularly advantageous in the post-KPG world, a period marked by fluctuating temperatures and the gradual cooling of the planet. The evolution of fur also provided new avenues for sensory perception, with specialized hairs, like whiskers, evolving to detect vibrations and explore the environment. The respiratory system also underwent a dramatic transformation, enhancing the efficiency of oxygen uptake and supporting the high metabolic demands of endothermy. The evolution of the diaphragm, a muscular sheath separating the chest cavity from the abdomen, was a pivotal innovation. This powerful muscle, unique to mammals, allowed for more efficient breathing by actively expanding and contracting the lungs, increasing the volume of air inhaled and exhaled. This enhanced respiratory capacity provided the oxygen needed to fuel the high metabolic rates associated with endothermy. Think of the rapid breathing of a small shrew, its diaphragm working tirelessly to fuel its high-energy lifestyle. Further refinements in the respiratory system involved the evolution of nasal turbinates. These thin, scroll-like bones, lining the nasal passages, dramatically increased the surface area for interacting with inhaled and exhaled air. Acting like miniature heat exchangers, the turbinates warmed and humidified incoming air, protecting delicate lung tissues from cold, dry air. Crucially, they also recovered heat and moisture from outgoing air, conserving precious water and energy, a particularly important adaptation in a world recovering from a mass extinction and experiencing fluctuating climates. The efficiency of this system is evident in the ability of desert-dwelling mammals, like camels, to conserve water in arid environments. The circulatory system, responsible for transporting oxygen and nutrients throughout the body, also underwent significant changes to support the high metabolic demands of endothermy. The four-chambered heart, a hallmark of mammalian physiology, evolved to completely separate oxygenated blood from deoxygenated blood. This separation, a major improvement over the three-chambered hearts of reptiles, ensured that oxygen-rich blood was efficiently delivered to tissues and organs, fueling the high metabolic rates needed for endothermy and sustained activity. This efficient circulatory system is what allows a cheetah to sprint at incredible speeds, its muscles fueled by a constant supply of oxygenated blood. Changes in blood composition further enhanced oxygen delivery. Mammalian red blood cells, unlike those of other vertebrates, lack a nucleus. This unique feature allows for more hemoglobin, the oxygen-carrying molecule, to be packed into each cell, increasing the blood's oxygen-carrying capacity. Furthermore, mammalian red blood cells are biconcave, a shape that increases their surface area and facilitates rapid oxygen diffusion. These adaptations, subtle but significant, further enhance the efficiency of the circulatory system, supporting the high metabolic demands of endothermy. These physiological adaptations, acting in concert, opened up a world of ecological possibilities for mammals. Nocturnal activity, a legacy of their Mesozoic ancestors, became a viable strategy, allowing mammals to exploit the cover of darkness for hunting and foraging while avoiding diurnal predators. The development of heterodont dentition, with specialized teeth for different functions, allowed mammals to diversify their diets, exploiting a wider range of food sources. Insectivores, like shrews, evolved sharp teeth for capturing and crushing insects, while herbivores, like deer, developed molars with high crowns for grinding tough vegetation. Carnivores, like wolves, evolved powerful canines for tearing flesh. This dietary diversification, coupled with the high metabolic rates enabled by endothermy, allowed mammals to thrive in a variety of habitats and occupy new ecological roles. The evolution of these physiological traits is inextricably linked to the diversification of mammals into various forms and lifestyles. Body size, influenced by factors like resource availability and climate, played a significant role in shaping these physiological systems. Larger mammals, like elephants, evolved slower metabolic rates and lower heart rates compared to smaller mammals, like shrews, reflecting the scaling of physiological processes with body size. Bergmann's rule, the observation that animals tend to be larger in colder climates, reflects the selective pressure for increased body size to conserve heat, particularly in the cooler environments of the late Cenozoic. The evolution of the mammalian placenta, a specialized organ connecting the developing fetus to the uterine wall, enabled prolonged gestation and the birth of more developed young, a reproductive strategy that fueled the diversification of placental mammals. The story of mammalian evolution in the Cenozoic is a testament to the interplay between physiological innovation and environmental context. Endothermy, fur, an efficient respiratory system, and a powerful circulatory system provided the foundation for mammalian success in a world recovering from a mass extinction event. These adaptations, coupled with changes in body size, dentition, and reproductive strategies, allowed mammals to occupy new niches, diversify their diets, and radiate into the remarkable array of forms we see today. The furry and warm-blooded creatures that inherited the Earth from the dinosaurs were not simply fortunate survivors. They were evolutionary triumphs, their bodies and physiologies sculpted by the relentless forces of natural selection to thrive in a new world. Chapter 5. The Gut's Microbial Garden, a co-evolutionary tale. The previous chapters have painted a vivid picture of the mammalian journey, from the synapsid shadows to the rise of the furry and warm-blooded. But this saga is incomplete without exploring a hidden world within mammals, a world teeming with trillions of microscopic partners, the gut microbiota. This chapter delves into the fascinating co-evolutionary tale of mammals and their gut microbes, a symbiotic partnership that has shaped digestion, immunity, and even behavior. The mammalian gut is not merely a digestive tube. It's a bustling metropolis, a microbial garden teeming with bacteria, archaea, fungi, and other microorganisms. This complex community, collectively known as the gut microbiota, plays a crucial role in mammalian health and evolution. Scientists have employed innovative techniques, such as network-based analysis of bacterial 16S rRNA gene sequences, to unravel the intricate relationships within this microbial world. These analyses, drawing on fecal samples from a wide range of mammals living in diverse environments, from zoos to the wild, reveal a complex interplay between host diet, phylogeny, and microbial diversity. Imagine a network where each node represents either a microbial genus or a mammalian host. Connections between these nodes represent the presence of a particular microbe in a specific host. The resulting network map, a visual representation of the gut microbial landscape, reveals fascinating patterns. For instance, herbivores with their plant-based diets tend to cluster together, their nodes connected by a shared set of microbial partners specialized in breaking down tough plant matter. Carnivores, on the other hand, form their own distinct cluster, their gut microbiota reflecting their meat-based diets. Omnivores, occupying a middle ground, exhibit a more diverse microbiota, reflecting their varied diets. This network-based approach allows researchers to map gut microbial community composition onto mammalian phylogeny and diet, revealing the co-evolutionary dance between host and microbe. Another powerful tool for understanding microbial communities is the uni-frac distance. This metric measures the evolutionary distance between microbial communities based on the branching patterns of their phylogenetic trees. Smaller uni-frac distances indicate greater similarity between communities, reflecting a shared evolutionary history. Studies using uni-frac distances have confirmed the influence of host species on gut microbiota composition. Conspecific hosts, even when living in separate locations, tend to have more similar gut microbiotas than hosts of different species. For example, red pandas housed in different zoos still share a more similar gut microbiota than they do with other animals in the same zoo. This underscores the strong influence of host phylogeny on the composition of the gut microbial community. The gut microbiota and its mammalian host are not merely cohabitants. They are active participants in a co-evolutionary dance, each shaping the other's destiny. The microbiota provides essential metabolic functions that mammals have not evolved on their own. For example, the microbiome encodes enzymes that break down complex carbohydrates, like those found in plant cell walls, releasing energy that the host can absorb. This microbial assistance is particularly crucial for herbivores, like cows and giraffes, whose diets consist primarily of plant matter. The gut microbiota also synthesizes essential vitamins, like vitamin K and certain B vitamins, that the host cannot produce on its own. In return, the host provides the microbiota with a stable environment and a constant supply of nutrients, a mutually beneficial arrangement. The gut microbiota also plays a crucial role in training the mammalian immune system. Early in life, the developing immune system learns to recognize and tolerate beneficial microbes while mounting defenses against harmful pathogens. This microbial education is essential for preventing autoimmune diseases and maintaining a healthy balance within the gut ecosystem. Intriguingly, the gut microbiota may even influence host behavior. Studies have suggested links between gut microbes and anxiety, depression, and even social interactions. The mechanisms underlying these gut-brain connections are still being explored, but the evidence suggests a complex interplay between the gut microbiome and the nervous system. The diversity of microbial communities across different mammalian species and diets is astounding. Primates, our closest relatives, offer a fascinating case study. Omnivorous primates, like chimpanzees and humans, exhibit a diverse gut microbiota, reflecting their varied diets. Folivorous primates, like colobus monkeys, have a specialized microbiota adapted for digesting leaves. Even within primates, unusual cases exist. The giant panda and red panda, both members of the order Carnivora, have evolved herbivorous diets. Their gut microbiota, surprisingly, resembles that of herbivores, reflecting their adaptation to a plant-based diet. This demonstrates the remarkable flexibility of the gut microbiota and its ability to adapt to the host's dietary preferences. The concept of co-evolution, the reciprocal adaptations occurring between interacting species, is beautifully illustrated by the gut microbiome. When a mammalian species adopts a new dietary niche, its gut microbiota undergoes corresponding changes. For example, the expansion of C4 grasslands during the Quaternary period led to the evolution of high-crowned teeth and longer gut retention times in herbivores that switched to a C4 plant-dominated diet. This adaptation to a lower-quality forage also impacted their gut microbiota, driving the evolution of microbial communities specialized in digesting these C4 plants. This intricate interplay between host and microbe is a key driver of diet-driven speciation, the formation of new species through dietary adaptations. Studying the gut microbiome presents unique challenges. Co-housing, the practice of keeping multiple animals together, can influence the gut microbiome, potentially confounding the effects of diet and phylogeny. To account for this, researchers often compare gut microbiotas of conspecific hosts living in separate locations. These studies have revealed remarkable consistency in gut microbiota composition within species, even when individuals are geographically separated. This suggests that host genetics and phylogeny play a dominant role in shaping the gut microbiome, even outweighing environmental influences. The human gut microbiota, it turns out, is typical of omnivorous primates. Our microbial partners reflect our evolutionary history as omnivores, equipped to digest a variety of food sources. However, modern lifestyles characterized by processed foods and reduced exposure to diverse microbial environments may be altering our microbial ecology. Understanding the impacts of these modern lifestyle factors on our gut microbiota is an area of active research, with implications for human health and well-being. The study of the gut microbiome has revealed a hidden world within mammals, a world of microbial partnerships that has shaped our evolution in profound ways. From aiding in digestion and training our immune systems to potentially influencing our behavior, the gut microbiota is an integral part of the mammalian story. This co-evolutionary tale, a testament to the interconnectedness of life, underscores the importance of considering the microbiome not as a separate entity, but as a fundamental component of the mammalian organism. The diversity of microbial communities across mammalian species reflects the diversity of diets and lifestyles, a testament to the adaptability of both host and microbe. And as we continue to explore this microbial garden within, we are sure to uncover even more fascinating chapters in the ongoing saga of mammalian evolution. Chapter 6. The Placenta's Embrace, a Story of Maternal Evolution. The gut's microbial garden, a bustling ecosystem within, has played a crucial role in mammalian evolution, shaping digestion, immunity, and even behavior. But the story of mammalian reproduction, particularly in placental mammals, takes an even more dramatic turn with the evolution of the placenta, a remarkable organ that transforms the maternal experience. This chapter delves into the placenta's embrace, exploring its profound impact on maternal evolution and its role in shaping the reproductive strategies of the most diverse group of mammals on Earth. The placenta, a temporary organ unique to placental mammals, connects the developing fetus to the uterine wall, creating a lifeline for nutrient and gas exchange. It's a remarkable feat of evolutionary engineering, a testament to the intimate dance between mother and offspring. But the placenta is not a monolithic structure. It exists in a variety of forms, each reflecting the unique evolutionary history and reproductive strategies of different mammalian lineages. The degree of placental invasiveness, the extent to which the placenta penetrates the uterine wall, is a key distinguishing feature. Imagine the different types of placentas as variations on a theme. At one end of the spectrum lies the epitheliocorial placenta, found in mammals like horses and pigs. In this type, the maternal and fetal tissues remain relatively separate, with 6 distinct layers separating the maternal and fetal bloodstreams. This less invasive placenta limits the transfer of certain molecules, such as antibodies, between mother and fetus, but it also reduces the risk of immune rejection of the fetus. Horses, for example, with their long gestation periods and relatively mature offspring at birth, benefit from this type of placenta, which provides ample time for fetal development while minimizing the risk of immune complications. At the other end of the spectrum lies the hemicorial placenta, found in humans and other primates. Here, the placental barrier is reduced to just 3 layers, allowing for a much more intimate connection between maternal and fetal blood. This highly invasive placenta facilitates the efficient transfer of nutrients and antibodies, crucial for the development of a complex fetal immune system. However, this close contact also increases the risk of immune reactions and complications like preeclampsia. Humans, with their large brains and extended period of postnatal development, benefit from the hemicorial placenta's ability to provide the fetus with a rich supply of nutrients and immune protection. Between these two extremes lie other placental types, each with its own advantages and disadvantages. The endotheliocorial placenta, found in carnivores like dogs and cats, represents an intermediate level of invasiveness, with the placental tissues eroding some, but not all, of the maternal uterine layers. The syndesmocorial placenta, found in ruminants like cows and sheep, is even less invasive, with the maternal epithelium remaining intact. This diversity of placental types reflects the remarkable adaptability of mammalian reproduction, with each lineage fine-tuning the placental connection to suit its unique reproductive strategy. The evolution of the placenta was not merely a matter of structural modifications, It involved a profound rewiring of the mammalian genome. Thousands of genes, originally performing functions in other tissues, were recruited and repurposed for placental function. This genetic overhaul was orchestrated by transposons, mobile genetic elements sometimes referred to as jumping genes. These genomic nomads, by inserting themselves into new locations in the genome, can rewire gene regulatory networks, turning genes on or off in new contexts. One striking example of transposon-mediated gene recruitment involves the synapsids. These genes, derived from ancient retroviruses that integrated into the mammalian genome, play a crucial role in the formation of the syncytiotrophoblasts, a specialized layer of the placenta responsible for nutrient and gas exchange. Synapsids promote cell fusion, allowing for the formation of a continuous layer of cells that facilitates efficient transfer of resources between mother and fetus. This retroviral legacy, once considered a genomic parasite, has become an essential component of mammalian reproduction. Other genes, involved in immune regulation, hormone signaling, and vascular development, were also recruited for placental function. The endometrium, the lining of the uterus, underwent a dramatic transformation, evolving to support placental attachment and development. Genes involved in cell adhesion, tissue remodeling, and immune tolerance were upregulated in the endometrium during pregnancy, creating a receptive environment for the developing placenta. This intricate interplay between placental and endometrial genes underscores the co-evolutionary nature of placental development, a delicate balance between maternal and fetal tissues. The evolution of the placenta unlocked a new era in mammalian reproduction, freeing mammals from the constraints of egg-laying and ushering in the age of live birth. This transition brought with it a suite of advantages. Live birth provided a protected environment for fetal development, shielding the developing offspring from predators and environmental hazards. It also allowed for extended maternal care, with the mother providing nourishment and protection even after birth through lactation. This extended care, coupled with the placenta's ability to provide a constant supply of nutrients, allowed for the development of larger brains and more complex nervous systems, contributing to the remarkable cognitive abilities of many placental mammals. The evolution of the placenta is intricately linked to other key mammalian adaptations, such as lactation and endothermy. Lactation, the production of milk by mammary glands, provides a rich source of nutrients and antibodies for the newborn, further enhancing offspring survival. Endothermy, the ability to generate and regulate internal body temperature, allows mothers to maintain a stable environment for fetal development and provide warmth for their young after birth. These traits, working in concert, created a powerful synergy that fueled the diversification of placental mammals. Even within placental mammals, a remarkable diversity of reproductive strategies exists, a testament to the adaptability of the placenta. Gestation length, the duration of pregnancy, varies widely, from the short gestation of mice and rats to the extended pregnancies of elephants and whales. Litter size, the number of offspring produced in a single birth, also varies dramatically, from the single offspring of primates and whales to the large litters of rodents and some carnivores. The level of maternal care, ranging from the minimal care provided by some rodents to the extensive parental investment of primates and whales, further reflects the diversity of reproductive strategies. Consider the extremes of reproductive diversity in mammals. The echidna, an egg-laying monotreme, lays a single leathery egg that hatches into a tiny, underdeveloped young. The newborn is then carried in the mother's pouch, where it nurses and completes its development. The kangaroo, a marsupial, gives birth to an even more underdeveloped young, which crawls into the mother's pouch and attaches to a nipple, remaining there for several months. These unique reproductive strategies, while different from placental mammals, highlight the remarkable diversity of mammalian reproduction. The placenta, a hallmark of placental mammals, has played a pivotal role in shaping maternal evolution and the reproductive success of this diverse group. Its ability to provide a protected environment for fetal development, facilitate nutrient and gas exchange, and mediate maternal-fetal communication has unlocked new evolutionary pathways, leading to the remarkable diversity of mammalian reproductive strategies we see today. From the intimate embrace of the hemicorial placenta to the less invasive epitheliocorial placenta, the placenta has adapted to the unique needs of each lineage, shaping the maternal experience and driving the diversification of placental mammals. Chapter 7. The whispering brain, the evolution of mammalian cognition. From the dimly lit forests of the Carboniferous to the bustling ecosystems of today, the mammalian brain has undergone an extraordinary transformation, a journey of expansion, reorganization, and the emergence of new connections. This whispering brain, the seat of mammalian cognition, reflects the remarkable adaptation of this diverse group to a constantly changing world. Our story begins with the earliest mammals, creatures of the night, whose world was primarily defined by scent. Their olfactory bulbs, the brain regions responsible for processing smells, were proportionally large, reflecting their reliance on this crucial sense for navigating the darkness, finding food, and detecting predators. The neocortex, the seat of higher cognitive functions, was relatively small, a mere whisper compared to the olfactory symphony. This emphasis on olfaction is reflected in the fossil record of early mammalia forms, like Morganucodon, whose skulls show enlarged nasal cavities and complex turbinates, thin bony structures within the nose that increase the surface area for detecting scents. As mammals diversified, venturing into new ecological niches and exploring new lifestyles, the brain underwent a dramatic transformation. The neocortex, once a minor player, began to expand, its layered structure providing the neural real estate for increasingly complex cognitive functions. This expansion was not uniform. Different brain regions evolved in response to sensory specializations. Primates, with their enhanced visual capabilities, developed expanded visual cortices, allowing them to process the rich visual information of their arboreal world. Echolocating bats, on the other hand, developed expanded auditory cortices, processing the intricate echoes used for navigation and hunting in the darkness. This mosaic of brain evolution, with different regions specializing in different functions, reflects the remarkable adaptability of mammals. The neocortex, the defining feature of the mammalian brain, deserves closer examination. Unlike the three-layered dorsal cortex of reptiles and birds, the mammalian neocortex boasts a six-layered structure, a complex architecture that provides the neural substrate for higher-level cognitive functions. This layered structure, with distinct cell types and connections in each layer, allows for sophisticated information processing, enabling mammals to learn, remember, and make decisions with greater flexibility and complexity. In reptiles and birds, many of these higher-level functions are performed by the dorsal ventricular ridge, DVR, a structure not found in mammals. The expansion and elaboration of the neocortex in mammals, coupled with the reduction of the DVR, represents a fundamental shift in brain organization, a key innovation in mammalian evolution. This neocortical expansion is evident in the fossil record of later mammals, with larger brain sizes relative to body size compared to their early ancestors. Brain connectivity, the intricate network of connections between different brain regions, also underwent significant changes during mammalian evolution. The corpus callosum, a thick bundle of nerve fibers connecting the two cerebral hemispheres, emerged as a defining feature of placental mammals. This structure, facilitating communication and coordination between the two halves of the brain, allowed for greater integration of information and enhanced cognitive abilities. The absence of a corpus callosum in monotremes and marsupials suggests that this structure evolved after these lineages diverged from placental mammals, further highlighting the unique evolutionary trajectory of this diverse group. The basal ganglia, ancient structures involved in motor control and reward processing, also underwent modifications in mammals. These modifications, involving changes in connectivity and neurochemistry, are linked to the evolution of more complex motor behaviors, enhanced reward learning, and the development of sophisticated social interactions. The basal ganglia's role in reward processing, for example, is thought to have played a crucial role in the evolution of parental care, a defining characteristic of mammals. The evolution of the mammalian brain is inextricably linked to the development of complex behaviors. Increased brain size and complexity, particularly in the neocortex, facilitated social interactions, communication, and cooperative behaviors. The emergence of diurnal behavior in some mammals, a shift from the nocturnal lifestyle of their ancestors, is thought to be related to changes in brain structure and function, particularly in the visual system and the circadian clock. These behavioral changes, in turn, likely drove further brain evolution, creating a positive feedback loop that propelled the development of mammalian intelligence. The diversification of feeding habits, from insectivory to herbivora and carnivory, also played a role in shaping brain evolution, driving the development of specialized sensory systems and cognitive abilities for finding and processing different food sources. The ability of some mammals, like primates, to use tools and solve complex problems is a testament to the remarkable cognitive abilities enabled by the mammalian brain. The evolution of mammalian cognition is a complex and multifaceted story, a tale of adaptation and innovation. From the olfactory world of early mammals to the complex cognitive abilities of modern humans, the mammalian brain has undergone a remarkable journey, shaped by the relentless forces of natural selection. The whispers in our genes, the symphony of bones and teeth, the physiological innovations of endothermy and lactation, the co-evolutionary dance with gut microbes, and the placental embrace of maternal care, all have played their part in shaping the mammalian brain and the remarkable cognitive abilities that define this diverse and successful group of animals. Chapter 8. The Social Fabric: How Cooperation Shaped Mammalian Destiny. The previous chapters have explored the remarkable journey of mammals, from their synapsid origins to the development of their unique physiology and intricate brains. But the mammalian story takes another fascinating turn with the emergence of complex social behaviors, a defining characteristic of many species. This chapter delves into the social fabric of mammalian life, exploring how cooperation, communication, and group living have shaped their destiny and contributed to their evolutionary success. Social behavior, encompassing the myriad interactions between individuals within a species, has profoundly influenced mammalian evolution. Many mammals, from the tiny meerkat to the massive elephant, live in groups, ranging from small family units to vast herds. This group living, a hallmark of mammalian sociality, offers a suite of advantages that have driven its evolution and shaped the destiny of many species. Consider the meerkat, a small mongoose native to the arid plains of southern Africa. These charismatic creatures live in tight-knit social groups, or mobs, of up to 50 individuals. Their social structure, a complex hierarchy with a dominant breeding pair and a network of helpers, is built on cooperation. Meerkats exemplify the power of cooperative vigilance. While some members forage for insects and other small prey, others stand guard, their bodies erect, scanning the horizon for predators. At the first sign of danger, the sentry emits a series of sharp alarm calls, alerting the mob to the threat. This cooperative vigilance, a key benefit of group living, enhances predator detection and allows for a rapid, coordinated response, increasing the survival chances of the entire group. Similar vigilance behaviors are seen in a variety of mammalian groups, from the vast herds of wildebeest on the African savanna, where individuals take turns grazing and watching for lions, to the close-knit packs of wolves in the northern forests, where sentinels guard the den while others hunt. Communication, the ability to transmit information between individuals, is the cornerstone of complex social interactions. Mammals have evolved a dazzling array of communication strategies, from the scent marking of territories by wolves to the elaborate songs of humpback whales. These communication systems, tailored to the specific needs and sensory capabilities of each species, facilitate coordination, social bonding, and the transmission of vital information. The songs of humpback whales, complex and haunting melodies that can travel for miles through the ocean depths, are a testament to the power of acoustic communication. These songs, primarily produced by males during the breeding season, serve multiple purposes. They attract potential mates, advertise the singer's fitness, and may even play a role in establishing dominance hierarchies. The intricate structure of these songs, with repeating phrases and evolving patterns, suggests a sophisticated level of vocal control and learning. Other whales, like dolphins, use a different form of acoustic communication: echolocation. By emitting high-frequency clicks and listening for the returning echoes, dolphins can navigate murky waters, locate prey, and even distinguish between different types of objects. This biological sonar, a marvel of evolutionary adaptation, allows dolphins to see with sound, a testament to the power of communication to shape sensory perception. Cooperation, the act of working together towards a common goal, is another hallmark of mammalian social behavior. Cooperative hunting, where individuals coordinate their efforts to capture prey, is a prime example. African wild dogs, highly social canids known for their coordinated hunting strategies, demonstrate the power of teamwork. These skilled hunters work together to pursue and bring down large prey, like zebras and wildebeest, that they could not capture alone. Their coordinated movements, often led by a dominant alpha pair, allow them to isolate and exhaust their prey, increasing their hunting success. Similar cooperative hunting strategies are seen in other mammalian carnivores, from the pride of lions encircling a buffalo to the pack of wolves coordinating their attack on an elk. Cooperative breeding, where individuals other than the parents help raise young, is another form of cooperation that has shaped mammalian societies. Meerkats, once again, provide a compelling example. In meerkat mobs, helpers contribute to pup rearing by providing food, guarding the young from predators, and even teaching them essential survival skills. This cooperative breeding strategy enhances the survival and development of offspring, increasing the overall reproductive success of the group. Wolves, another highly social species, also exhibit cooperative breeding. Helpers, often older siblings or non-breeding adults, assist in raising the alpha pair's pups, providing food, protection, and social interaction. This cooperative effort strengthens the pack and ensures the survival of the next generation. The evolutionary drivers of sociality in mammals are diverse and complex, reflecting the interplay of ecological pressures and social opportunities. Predation pressure, the constant threat of being eaten, is a powerful force promoting group formation. Herd behavior in ungulates, like zebras and wildebeest, provides protection through increased vigilance and the dilution of individual risk. A single zebra in a herd is less likely to be targeted by a predator than a solitary individual. Sociality can also enhance foraging success. Cooperative hunting, as we've seen in African wild dogs, allows predators to capture larger prey, providing more food for the group. Primates, like chimpanzees, often share information about food sources, increasing foraging efficiency for the entire troop. Social learning and cultural transmission, the passing of knowledge and skills between generations, are further benefits of complex mammalian societies. Chimpanzees, our closest living relatives, provide remarkable examples of tool use passed down through generations. Young chimpanzees learn from their elders how to use sticks to extract termites from mounds or stones to crack open nuts, demonstrating the power of social learning to shape behavior. Whales, like orcas, have distinct vocal dialects that vary between pods, suggesting a form of cultural transmission of vocal patterns. The relationship between sociality and evolutionary rate is intriguing. More sociable mammals tend to evolve faster than solitary ones, a phenomenon linked to the intense selective pressures associated with social interactions. Competition for mates and resources within a social group drives the evolution of traits related to dominance, communication, and cooperation. The need for effective communication in a social setting favors the evolution of complex signaling systems, from the elaborate songs of whales to the intricate facial expressions of primates. The constant need to cooperate and compete within a social group creates a dynamic environment that accelerates evolutionary change. The rapid evolution of primate communication signals, from the simple alarm calls of monkeys to the complex vocalizations of chimpanzees, illustrates this phenomenon. The evolution of complex social structures in cetaceans, from the matrilineal pods of orcas to the fission fusion societies of dolphins, is another example of rapid evolutionary change in social mammals. The constant need to navigate social hierarchies, cooperate in hunting and foraging, and care for young creates a selective pressure cooker that drives rapid evolutionary innovation. The social fabric of mammalian life is a tapestry woven from threads of cooperation, communication, and competition. Group living, a hallmark of many mammalian species, provides protection from predators, enhances foraging success, and fosters social learning. Communication, in its diverse forms, allows individuals to coordinate their actions, bond with others, and transmit vital information. Cooperation, from cooperative hunting to cooperative breeding, strengthens social bonds and enhances the survival of the group. These social behaviors, shaped by ecological pressures and social opportunities, have played a pivotal role in mammalian evolution, contributing to the remarkable diversity and success of this extraordinary group of animals. Chapter 9. Giants in the Aftermath. mammalian titans of the Cenozoic. The asteroid impact that marked the end of the Cretaceous period, 66 million years ago, brought about a dramatic reshaping of life on Earth. While the dinosaurs, the dominant terrestrial vertebrates for over 150 million years, vanished, a new era dawned, the Age of Mammals. The Cenozoic era witnessed not only the diversification of mammals into a vast array of forms but also the rise of true giants, colossal creatures that dwarfed their modern relatives and dominated the planet's ecosystems. The extinction of the dinosaurs created a vast ecological void, a world of opportunities for the surviving mammals. New habitats opened up, previously inaccessible food sources became available, and the competitive landscape was transformed. This ecological release, coupled with a warming climate and the spread of lush forests, provided the perfect conditions for mammalian diversification and the evolution of gigantism. Imagine a world where dense forests extended even to the poles, teeming with vegetation and providing ample sustenance for a burgeoning population of mammals. The fossil record of the early Cenozoic tells a tale of rapid evolution and dramatic increases in body size. Within a few 100,000 years after the KPG extinction, mammals had already doubled their pre-extinction size, reaching weights of up to 7 kg. This initial growth spurt was followed by an even more dramatic increase, with some mammals reaching weights of 15 kg within 300,000 years. This rapid expansion was fueled by the flourishing plant life, with the emergence of the bean family, or legumes, about 700,000 years after the KPG event, providing a new source of high-protein food that further fueled the evolution of larger mammals. The evolution of these mammalian giants was not a uniform process. Different lineages, on different continents, responded to the ecological opportunities in their own unique ways. One of the most iconic examples of Cenozoic gigantism is Endothermy, a hornless rhinoceros-like herbivore that roamed the forests of Eurasia during the Oligocene epoch, about 34 million years ago. This colossal creature, standing about 18 feet high at the shoulder and weighing an estimated 17 tons, represents the largest land mammal that ever lived. Its long neck and high-crowned molars allowed it to browse on the leaves of tall trees, much like a modern-day giraffe, but on a scale that dwarfs even the largest extant land mammals. Endothermy was not alone in its gigantism. The Cenozoic saw the rise of giant mammals in various lineages. Endothermy, a bizarre herbivore with 6 bony horns on its skull, roamed North America during the Eocene. Arsinoitherium, an African ungulate with a pair of massive horns above its nose, lived during the Oligocene. Dinotherium, a relative of modern elephants with downward curving tusks on its lower jaw, inhabited Eurasia and Africa during the Miocene and Pliocene. These examples, just a glimpse into the diversity of Cenozoic megafauna, showcase the remarkable evolutionary potential of mammals. The study of these extinct giants relies heavily on the fossil record. Teeth, the hardest parts of the mammalian body, are often the best preserved remains, providing invaluable insights into the diets and lifestyles of these ancient creatures. The high-crowned molars of Endothermy, for example, tell us that it was a browser, feeding on tough vegetation. The sharp teeth of Andrewsarchus, a wolf-sized carnivore from the Eocene of Asia, suggest a predatory lifestyle. Skeletal structures, when preserved, offer further clues. The robust limb bones of Coryphodon, an Eocene herbivore, indicate a large, heavily built animal adapted for traversing dense forests. The rise of these mammalian giants was not without its challenges. Increased body size requires more resources, making these animals vulnerable to environmental changes. The cooling climate of the late Cenozoic, coupled with shifts in vegetation, likely contributed to the decline and extinction of many giant mammals. As grasslands expanded and forests contracted, many of these large herbivores, adapted to browsing on trees and shrubs, faced dwindling food supplies. The changing climate also brought about more seasonal variations, with harsher winters and drier summers, further stressing these giant mammals. The emergence of new predators, like the large carnivores that evolved alongside these giants, also played a role in their decline. The study of maximum body size in terrestrial mammals has led to several theories about the factors that limit growth. Resource availability, the amount of food and water available to sustain a large body, is a key constraint. Climate, particularly temperature, also plays a significant role. Larger animals conserve heat more efficiently, giving them an advantage in colder climates, as exemplified by Bergmann's rule. Land area, the amount of space available to support a population of large animals, is another important factor. The interplay between these ecological and evolutionary processes and abiotic factors like temperature and land area ultimately determines the upper limits of mammalian body size. The fossil record provides a glimpse into a world dominated by mammalian megafauna, a testament to the remarkable adaptability and evolutionary potential of mammals. These giants, like Indricotherium, Uintatherium, and Dinotherium, occupied ecological roles similar to those of large dinosaurs, shaping the ecosystems of the Cenozoic. Their rise and fall, a story etched in bone and stone, provide valuable insights into the interplay between evolution, ecology, and environmental change. Their extinction, a reminder of the fragility of even the most dominant life forms, serves as a cautionary tale about the potential impacts of climate change and habitat loss on today's biodiversity. The transition from the small, nocturnal mammals of the Mesozoic to the giant forms of the Cenozoic is a remarkable chapter in mammalian evolution. The extinction of the dinosaurs, a catastrophic event that wiped out the ruling reptiles, created the ecological space for mammals to diversify and thrive. The Cenozoic giants, inheriting this new world, represent a testament to the resilience and adaptability of mammals, their ability to seize opportunities and evolve into a stunning array of forms, from the tiny shrew to the colossal Indricotherium. Their story, a tale of triumph and tragedy, continues to unfold as we delve deeper into the fossil record and unravel the secrets of mammalian evolution. The evolution of mammals is a captivating story of adaptation and diversification spanning hundreds of millions of years. From their humble beginnings as small, nocturnal creatures in the shadow of dinosaurs, mammals rose to become the dominant terrestrial vertebrates, radiating into a breathtaking array of forms, from tiny shrews to massive whales. Several key transitions fueled this remarkable journey. The evolution of an erect posture and specialized teeth allowed for greater agility and exploitation of diverse food sources. The transformation of the jaw and ear enhanced hearing, particularly crucial in the nocturnal world of early mammals. The development of endothermy, fur, and a four-chambered heart provided the physiological foundation for sustained activity and adaptation to a wider range of environments. The evolution of the placenta in placental mammals revolutionized reproduction, allowing for prolonged internal development of the fetus and the birth of more developed young. The mammalian brain, with its expanding neocortex, enabled complex cognitive abilities and sophisticated social interactions. These evolutionary innovations, intertwined with environmental changes and ecological opportunities, shaped the mammalian lineage. The extinction of the dinosaurs, a pivotal moment in Earth's history, opened up ecological space for mammals to diversify and thrive. The Cenozoic era witnessed not only the radiation of mammals into various niches but also the rise of mammalian giants, colossal creatures that dominated the planet's ecosystems. The co-evolution of mammals with their gut microbiota further shaped their digestive capabilities, immune systems, and even behavior. The intricate social fabric of mammalian life, with its emphasis on cooperation, communication, and group living, contributed to their evolutionary success. These findings have profound implications for our understanding of life on Earth. They illuminate the interconnectedness of organisms and their environment, the power of natural selection to shape form and function, and the remarkable adaptability of life in the face of change. The study of mammalian evolution also has practical applications. Understanding the genetic basis of mammalian traits can inform medical research and the development of new treatments for human diseases. Insights into mammalian social behavior can inform conservation efforts and the management of wildlife populations. Looking forward, the study of mammalian evolution promises even greater discoveries. Advances in genomics and molecular phylogenetics will continue to refine our understanding of mammalian ancestry and the timing of key evolutionary events. Further exploration of the gut microbiome will reveal the intricate connections between microbes and their mammalian hosts. Paleontological discoveries will unearth new fossils, providing glimpses into the lives of extinct mammals and filling in the gaps in our evolutionary narrative. What questions do these remarkable discoveries spark in you? Share your thoughts and comments below. We encourage you to like and subscribe for more explorations of the natural world. Suggest topics you'd like us to cover in future videos. Never stop questioning, never stop learning. The journey of discovery is an endless adventure, and the story of life on Earth is a tale worth telling, again and again.